Thank you very much again. And we will go back from US to Europe, having Marisa Papaluca from EMA, and she will present the vision of the European regulatory agencies. Please, Maria, take the floor.
Thank you very much. And I have to say, I have to congratulate with the previous speaker, because it was really speakers, actually, both of them. It was really very, very good presentations. So I kept myself very simple, because I think we have enough complexity in the kind of drugs we have. I would like to say, when it comes to bioequivalence studies and the opportunity to register in Europe generics, we have really just the difficulty to choose which one of the route we want, what kind of guideline we can follow, because this is such a wealth of information and opportunities to meet at the EMA level, at local level with the national competent authorities. And some of the work I will present later on today really is the results of work done in the two systems, the centralized and the decentralized system, and you will see that the kind of issues and the kind of discussion you have, the kind of guidance you can get is very much specular. So it's just a matter of choosing what is most appropriate for the fate of that particular product you want to register, and also the kind of penetration of the market you wish to explore. We have a lot of guidance, because I didn't prepare an overview of the existing guidance, because I think I made it last year. So we have some guidance for nanotechnology-based products, like follow-on for liposomal formulation, iron oxide particles. We had a reflection paper on the importance of the coating around nanoparticles and nanomaterials. And we have also a long string, which I didn't bring with me, I don't know by heart, of all specific, product-specific bioequivalence requirements for studies for new generics. And this was a work which was carried out. We didn't trumpet too much, I think, maybe too little, but based on input from the member states or which kind of generics they want to facilitate, because they already had a lot of contacts with sponsors. We produce, I think, more than 18 guidelines product-specific for bioequivalence. This was done under the leadership of the Pharmacokinetics Working Party, of the committees in the EMEA, and they're really very useful. A little bit different from what the FDA has done, but I think serve the purposes. Now, I would like to move more on what are the issues and current experience with the so-called non-biological complex drugs. This is to go ahead, I suppose. I suppose it's this way. No. It's the other way? No. Which way is it? Yeah, okay. So I think I will be very concise. I think the colleague from the FDA made a very splendid presentation on very specific technical aspects. I would say, as we don't have in Europe a definition for non-biological complex drugs as such, we can say that we are confronted with the complex molecular structures, and we have mixtures, we have a liposomal formulation with the various types of coated iron oxide nanoparticles, gels, and so forth. We have interesting, and also in this meeting there was some discussion about long-acting injectables, which really in themselves constitute a problem, not problem, but a challenge when it comes to the kinetics of the products, and complex drug-device combination, for example, drugs and sensors. And we have to remember that this can only increase as a complexity, because digitalization will see us with more and more mixture of chemical and digital elements, which will have an impact on each other. And then the synthetic peptides we see, and the so-called generics of non-biological complex drug, the one we dubbed as nanosimilars a few years ago. And I think we were quite visionary when we published this little article, because in fact today for complex non-biologicals, most of the time in Europe at least, we speak about how similar those products are to the originator. for the challenges, very quickly, characterization methods, of course, are a problem because not all methods are really well standardized, and the exposure to the characterization methods is very important. That happens at an early stage. So I can only second the previous speakers' invite to all innovators to come very early, especially if they intend to make a modification of a method already in place to adapt to the specific product. We have the so-called product and process-related molecular species, which is very interesting. Some of them are impurities, others are not impurities, are just molecular species which are derived from the manufacturing process and contribute to the mixtures. We have that it's very important to see at the active substance and components release kinetics. And again, as already said, is not just the so-called active, but also the other components of the, for example, of a nanosystem, which have an important role in safety and efficacy, as already explained. Also, it's very important to be aware that we try also to understand the relative contributions of the various components. When you have a complex drug, you don't ask for each individual component's specific contribution, but you have to explore whether there is a contribution, either in positive or negative. Because it's very rare that we have components which are so-called inert, passive. I still have to see these things, been in 35 years in this area. So, and then a specific conversation should take place, as already initiated by the first speaker, on the follow-on, also called similar products. So, what are the regulatory requirements? I mean, we have the legislation which guide us, is Article 10.3. We have the opportunity for, particularly for the generic similar of non-biological complex drugs, we know that we can rely in part on reference to results of preclinical tests and clinical tests of the reference product. We can use very much, and I will show you cases in which very much literature has been used as a replacement for own data. Then we have to make something very important as a statement here in this international conference. I mean, the reference products against which any comparability will have to be carried out for this kind of products should be a product which has been authorized in the European Union. So the bridge should be with with products which have an authorization in the European Union. And then, of course, depending on how, not how similar, but how different the follow-on is, and therefore the innovator should look at the differences. You can have a very vast range of requirements, even from a single rational explanation up to large battery studies. So again, I repeat that even for any study you intend to put in place for the comparability exercise, that should be with an EU-sourced RMP. Now, the good news is that really there is flexibility in market authorization submission. This is why it's so important that people developing either complex, these complex drugs or even follow-on of that, they should come very early. Normally, when it comes to the follow-on, and because in the previous meetings here we discussed a lot how a follow-on is authorized in Europe, normally it's a hybrid application, so there's a mixture of own data, literature data, and data from the reference product. And here I want to present two interesting cases. One is the synthetic teriparatide teva, authorized in 2017, which was developed in comparison with Forsteo, which was a teriparatide DNA recombinant derived product. So what we asked for that, and compare what we asked for the Brabio, the follow-on of Copaxone. Of course, we had a full drug substance dossier with all characterization, plus comparative analytical data with Forsteo. And at the end, the conclusion was that the two products, even though they were not absolutely identical, actually they were very similar. They were similar, they were comparable. There was an in vivo study in rats, and in particular, it was looked at no immunogenicity concerns were raised. And there was a lot of also supplementary information provided in the dossier derived from literature as the pharmacokinetic and the pharmacodynamic of teripathide is pretty known. And then there was a bioequivalence study versus Forsteo, and the interesting things, and I want to attract your attention on that, is not just the old-fashioned bioequivalence with 10 people captured somewhere, was a very well-designed study on 72 subjects in which not only the equivalence and the dynamics was studied, but also safety. And there was quite an important package for looking at the safety aspects as well. So then, and that's it. There was no requirement for additional clinical studies, and this was approved and available in a large number of member states. This was approved and nurtured, I would say, from a national competent authority, which was the reference competent authority, and then via the decentralized system taken on board in, I think it's 14 member states. If we look at what happened with glatiramer acetate, where we know there is a divergence on what we did in Europe and what the FDA did in their region, only 2 minutes left, so I will be very quick on that. The big difference is that there was a large physicochemical characterization test, battery tests. There were non-clinical tests, quite extensive, including immunogenicity. And then there was a substantial phase 3 clinical study, which was very, very carefully designed in collaboration with the regulators, and which actually could demonstrate a therapeutic equivalence in multiple sclerosis. We have to say, in that case, not only the substance was complex, but also the disease is very complex. So when you put the two things together, you have to be absolutely sure to be able to discriminate from where issues can emerge. So for a disease in which the actual mechanism of effect of the product is very complex, not just the product itself, but the way it acts on the disease. And the fact that the disease is quite an escaping disease. As you know, there is now a new taxonomy emerging for multiple sclerosis, because it looks like it's not one disease, it's many different diseases. And we know with the emerging taxonomy and in clinical care how difficult it is, especially if you are not very advanced stage, to discriminate between the various subforms of the condition. So certainly it is an important area. So what we have to do now in order to see whether we can avoid the discrepancies, because I think we had a very good reason. I think this was the reference member states was Netherlands. Again, 15 or 16 member states now have Brabio on the market. This was complex in all respects, but we would like to see in the future that if a company develops a complex drug in Europe, does not have to do something different than in the United States. So I take advantage of a few more minutes just to see the current, this is where we are, and now I tell you the trends. So in July 2016, we approached the FDA to understand their requirements for complex generics and to explore possibilities for global development. And this happened at a very high level, not at operational level. Then in June 2017, the discussion started with the European Commission, which is our legislator and our master, on how we can look from the regulatory point of view at criteria for acceptability of a global development of a complex hybrid. In parallel, in 2017, the new commissioner, of course I preached to the one already know everything, but actually was looking at how we can make it easier for people to have more opportunities of treatment. So especially in those particular areas where there is no competition, because there's one product which makes it for everything and for over, for everyone and for all. So there was quite an emphasis from the new commissioner on this aspect, which actually came at the same time where we were trying to find useful dialogue. So then what we have, where we are now in Europe, what we need to consider in this possibly new international dialogue. First, we don't have a legal definition. So we think it's timely to start having a conversation at the international level of sort of categorizing what complex products are, and it was very interesting what previous speakers presented. There's already an effort there. So we can start discussing, is possible to have a complex global development? And also, is possible to use a non-EEA approved products as a comparator? Let's assume that the product is developed completely in the United States. We will be able to accept the data even though the comparator is not a European one. These are questions we need to consider internally in Europe and then be prepared to discuss internationally. But there is a good news. We dubbed some nanotechnology-based products nanosimilar, and as you know, I've been sort of the mother of the biosimilar concept in Europe in 2004. And now I want to conclude this presentation with an interesting development which took place recently on the global development of biosimilar, because in 2014, the overarching guideline on similar biological medicinal product, the concept of acceptability of non-EU reference medicine was accepted. Of course, under certain stringent conditions, but this, I think, is the trend. So let's nurture and learn from the biosimilars, see how all the experience we had accrued in more than 10 years on almost 15 years on biosimilars. We have not seen any death under the bridge. We have not seen any special safety signal or lack of efficacy signal with all the biosimilar we had. So maybe we can learn and build on that for these new categories. And I'm ready for questions. Thank you very much. Sorry, I took some more minutes.
Thank you very much, Marisa, for as well an excellent presentation on trends. And there is some more than some light in the tunnel, that there are a tendency to go for that. Urgent question, Dan.
Dan. I know. Yeah. I knew I could not avoid it.
Sorry, but
I'm very happy.
You mentioned complex hybrids.
Yeah.
And that's a new term.
Yeah.
In the field. And Jon already in his introductory presentation made the point that the vocabulary and terminology is in this field is a gray area, and that we would like to sort of make sure that we get globally one sort of terminology box. And I know that hybrids exist, I know that complex exists, but the combination of this is new to me. So why did you come up with this new term?
Yes, this time is not my fault. For nanomedicine, I can take responsibility as well as for nanosimilar. For this one, it's not my fault. This is based on actually very heated discussions with the legal services. And when we speak about complex hybrid, we do not refer to the product. We refer to the package for the marketing authorization application for the ANDA. So it's a complex hybrid. So it's not a full dossier, it's in between, it's hybrid. You have own data, even big clinical studies, like is the case for the glatiramer's, and a lot of literature and data from a previous approved product. So this is a terminology which is not scientific and is not referred to the product, but to the complex hybrid regulatory submissions. So this is a sort of, the lawyers want to do something as well, and something interesting, I mean. I'm joking. No, I know that this is recorded, so I just have to qualify. These are personal comments, but I think they touch the point. It's not the very simple standard dossier you see for generics. For generics, you know very well the path. There are a certain number of studies that need to be done, the critical quality attributes to be defined, quality by design, and then you have the bioequivalence. And that one will be scrutinized and everything, okay? this is not the case for if you have a complex product for a complex condition. That will require an hybrid submission in the best case. But for example, I don't know when I will present that, maybe at the closed meeting, because some more confidential information. If you look at what we had, even the centralized system for new teriparatide, again, some of them, I think one of them came as a full dossier because they want to put something completely new. So it was not legally a full dossier, but was a huge dossier because they had the full pharmacodynamics in animal and humans, they had big studies. So this is why they say the basis Article 10.3 for generics. We don't have in the legislation, as for biosimilars, the terminology for NBCD. So we have to find a way to describe that for certain products you have a different submission expected. And again, the quicker, the earliest the people from generic industry come for conversation, either at the EMEA or at the national level, the better it is. But again, it's the two things, the complexity of the substance, of this construct of the system, and the complexity of the disease you intend to treat. These are explosive.
Okay, Asel, short.
Is it acceptable to comment on that? I really want to support Dan with his question, and of course it's understandable why you came to the conclusion to combine the complexity of the product and the hybrid application. I really know how difficult it is to come to a certain harmonization, but harmonization of terminology is a nightmare. It should be very easy. Why abbreviated new drug applications on the US and abridged application in Europe? Why pharmaceutical equivalence approach and essential similarity in Europe? It's, from a scientific point of view, not understandable. And my comment is, if you define this new terminology for this kind of drug, it is a nightmare to come to consensus with US FDA because they do not know hybrid application. And if you define that, this is an obstacle for harmonization in a field where it should be very easy as long as it's not written in stone on both sides. Therefore, I think Dan's comment is and you should reconsider that.
Message taken, and this will be reported, is not in the legislation as of yet. As you know, for DNA recombinant, we had to put the things in legislation, the similarity, the biosimilar kind of category. Yeah. For this one, we are not there. We are not going to open the legislation to put complex hybrid in, okay? But maybe it's a point to discuss further with FDA, with Canada, with China, with all the colleagues. But we have to find a way to signal to the sponsors that they will not get away with just a simple bioequivalent study, and that's it. Or even only literature, because some tries only to come only with literature and a tiny package for characterization. Come on. So, but anyway, thank you for the comment and will be taken on board. Yeah. Thanks a lot.
Thank you very much. We took a little more time for that, but I think that opened the question from the very end. I think very helpful to have that. And Dan, on the other hand, I think your book on the terms will never be ended. So we have to go into that. Now,